Hello, everybody, and welcome. My name is Janneke Schwanner, and I'm a postdoctoral researcher in the Department of Ecology and Evolutionary Biology at the University of California, Irvine. I'm happy to welcome you in a series of workshops on integrative organismal modeling of movement. These workshops have been sponsored by the National Science Foundation Division of Integrative Organismal Systems, with the goal of bringing together scientists at the intersection of organismal biology and physics. We aim to highlight the use of model-based and mathematical tools to address fundamental questions in organismal biology. We hope that these workshops will generate dialogue and collaborations to help both experimental and theoretical scientists make effective use of mathematical and modeling tools for understanding animal movement and behavior. This workshop will be recorded and shared as an online resource hosted through the UCI Center for Integrative Movement Sciences with the School of Biological Sciences. And with that, please welcome the rest of today's workshop speakers, Taylor Dick from the University of Queensland, Kisa Nishikawa from Northern Arizona University, and Surabhi Simha from Emory University. And then I will pass it on to the first speaker.
Thanks, Janneke, and thanks everyone for joining us as part one of our two-part series on multi-scale muscle modeling. So the plan for this 90-minute session is really to showcase different flavors of muscle models and to highlight their applications as well as some of their limitations. And we'll do so with a strong focus on experimental data. So what in vivo or ex vivo data can we collect? And how can we use this data as model inputs or to evaluate the predictions from our models? So just before we get started with introducing ourselves, we want to acknowledge the support from NSF as well as the incredible efforts of Monica, Kisa, and Brooke for facilitating this. So well done. It's really exciting to see this all come together. All right, so let's introduce ourselves. My name is Taylor Dick. I'm an academic at the University of Queensland in Brisbane, Australia. I did my PhD in Canada and my postdoc in the US before starting my lab here at UQ about 4 years ago. And really broadly speaking, my group is interested in understanding the neuromuscular and biomechanical mechanisms that underpin healthy and pathological movement. So more specifically, our current interests are in discovery science. I have a passion to understand the fundamental mechanisms of muscle and tendon through a combination of experimental and modeling approaches. And this is really what the bread and butter of I'll talk about today is. But my lab's also interested in understanding how muscle and tendon properties and motor function adapt to external challenges that might arise as a result of changes in body size, age, and disease, not just in humans, but particularly interested in scaling across different animals and different species of different sizes. And finally, we're interested in investigating and working closely with engineers to understand how wearable assistive devices such as exoskeletons and prosthetics can interact with biological tissues to augment, to perturb, or to restore movement. So that's a little bit about me. I'm excited to work with all of you. So over to you, Kisa.
Thanks so much. It's great to see so many people here, and I'm just excited to be a part of this group. So my name is Kisa Nishikawa. I'm a Regents Professor at Northern Arizona University. And well, I don't know, for seeming decades, I've been interested in muscle mechanics and really sort of trying to integrate from molecules to movement. So one of our main projects has been to investigate the role of titin in active muscles, and particularly the role of tunable spring properties of titin and how they might contribute to history-dependent force production. So over the past few years, we've also been working in my lab to improve predictive models of in vivo muscle function, and I look forward to talking more about that shortly.
Yeah, so I already introduced myself a little bit, but my name is Janneke Schwanner, and I'm a postdoctoral scholar in the neuromechanics lab at the University of California, Irvine. I'm generally interested in unsteady locomotion, anything from jumping and complex terrain navigation, locomotor mechanics and sensory control, in vivo and in situ muscle tendon dynamics, and I will tell you more about that during this workshop. And I'm also interested in inertial post-anal appendage use for aerial stability and maneuverability.
Hi, my name is Surabhi Simha. I'm a postdoctoral fellow at Emory University and Georgia Tech. And I did my PhD in Canada until like 2 years ago. And in that time, I studied motor control of how people move in familiar environments and new environments. And I focused a lot on energetics and mechanics of by measuring people as they walk and also developing whole body models. And I've been working during my postdoc on developing mechanistic muscle models that I can hopefully take and use in these whole body simulations and understand motor control. And hopefully today I'll talk about the mechanistic muscle models with you.
Brilliant. So now that we know a little bit about who we are, here's a bit of an outline for our tentative plan for this first workshop. So first, Sarabi will cover crossbridge models. Kisa will go into Titin clutch and hill type models with a focus on animals. I'll cover hill type models with a focus on human. Yannicka will cover some experimental methods, and then we'll have a period for questions and discussion at the end. I think you can post questions in the chat box throughout the talks as they come to mind. But other than that, I'll pass it over to Sarabi to kick things off.
Yeah, thank you, Taylor. Okay, so I'm going to hopefully try and explain how a crossbridge muscle model works. So I have a few learning objectives for this talk. First, I'd like to talk about what we want to model in muscles and why, where I'll have a focus on motor control and muscle transient force response. Then I'll spend some time discussing how to model muscle forces, where I'll very briefly touch upon the physiology and then spend some time on the math and the inputs and outputs in the crossbridge muscle model. Finally, I'll briefly discuss how we evaluate the model, and I'll focus on comparing key features from the model to experimental data. Okay, so why do we care about muscles? Because muscles are what has let us interact with the world. So briefly, amongst the many things that muscles can do, they generate forces. So anytime we push or pull or anything, this is the muscle forces that let us do that. So here is a schematic of a muscle being stretched and the corresponding force data during the stretch. Muscles also consume energy. And like I said, I spent a lot of my PhD measuring how people consume energy while they walk in different ways. And a lot of this energy is really being consumed by the muscles. And also, interestingly, muscles sense mechanics. So here's a schematic of a muscle spindle and some data from a 1A afferent that really reads from your spindle. Now, I'm interested in all of these and would like a muscle model that can capture all of these features. But for this talk, I'm going to focus on how muscles generate force and particularly focus on force during stretch because it is a useful example to try and explain the crossbridge model itself. So why care about muscle forces? This is because muscle forces can impact motor control. To explain this, here's a schematic of a person standing on a platform when the platform is quickly pulled forward. When this happens, the person falls backward and they have many mechanisms to stabilize themselves. I've grouped these mechanisms into two over here. One is the muscle intrinsic response that is contained wholly within the muscle and happens really quick at less than 50 milliseconds. And then you have the longer neuromechanical response that involves different parts of the nervous system and takes at least about 80 milliseconds. Now, here are data of what a person does during such a perturbation. This is the hip joint angle, and this red vertical line here corresponds to when the EMG comes on during this perturbation. And this happens at about 100 milliseconds, so it's really showing you this longer neuromechanical response. But then if we look at the hip joint torque, it comes on quite early at about 50 milliseconds. So to understand what's happening with this hip torque, let's look into a single muscle fiber. Now here is the length data when a single muscle fiber is being stretched over time, and this is the corresponding force data. And if you focus on this gray bar over here, you see that the force here rises quite quickly and goes pretty high. Now this is the transient muscle force response, and it corresponds to this hip torque that you see up here. And we think that this transient force is due to the muscle intrinsic response to stretch that I talked about earlier. And this transient force can affect motor control. As an example, this is an animation of a person standing on a platform when the platform moves backwards. And you can see that they do a pretty good job of stabilizing themselves. And now here is a video of a simulation of a model during the same perturbation. Now, this model consists of muscle models, and the muscle models do a good job of capturing the forces during the later phase, but the models don't include a transient force. And you see that the simulation mostly falls over. So we would like to model a muscle that predicts steady state and, importantly, transient forces as well. So the simulation that I just showed you, where it sort of falls, uses a traditional hill-type muscle model. Now, these are the type of models that are very commonly used in the field, and you have lots of variations of these. And I think both Kisa and Taylor will talk a lot more about this going in. Just the one point I want to focus on this slide is that these muscle models can really predict steady state forces. By this, what I mean is that the force in the model is derived based on steady state relationships of force as a function of length and force as a function of velocity. The models do have activation dynamics, so by steady state, I'm referring to the relationship to length. And to understand that, here is a simulation of a muscle. And in the bottom row, you see the muscle changing length and arriving at the new constant length. And in the top row, you see the corresponding force changes. Now, the force-length relationship in a Hill model really takes the force from this later part where the force has reached steady state. But as you can see with this red dotted box, it takes a while for the force to reach steady state, and this is your transient force. Now, the force-velocity relationship used in the Hill model is a little bit more complex to explain this way, but it also similarly depends on steady-state properties. But the Crossbridge model can fit a transient force response. And to show that, I bring back a similar data set as I showed a couple of slides earlier. So in the lower row, you see the muscle fiber length change as a function of time. And in the top row, you see the corresponding force changes. So once again, if you look at the very beginning of the initial stretch, you see that the force quickly rises up and then goes higher than it will throughout the rest of the trial. And over here in the top row, you can also notice that there is a black line, which is experimental data. And on top of that are these red circles, which is model simulation using a Crossbridge model, which quite well predicts the data. Now, the Crossbridge model is not the only model that can predict transient forces. And Kisa is actually going to come and talk about the Titin model, which captures different features of the features of the transient forces. But maybe an interesting point to think about here is the Hill model does quite well at capturing whole muscle features. But both the Crossbridge and Titin model that get at transient forces go quite deep into the muscle. So based on that, here's a really brief primer of the muscle itself. Again, I'm going to go by this very quickly, but Janiga is going to come in at the end and hopefully connect these abstract concepts into actual real muscles. So this is a schematic of a general muscle from a textbook. And you can see the muscle is connected to your bone using these passive tissues, the tendons and connective tissues. And this is your Hill model. But within the muscle, you have a lot other things going on. Particularly, you have these single cells called muscle fibers. And these muscle fibers contain proteins called these muscle fibers contain proteins called myofilaments, and I'm really going to quickly focus into these myofilaments that is present within the muscles. And the reason for the focus on the myofilament is because this is where the force is generated within a muscle. Now at the top, you see in yellow the actin filament, and below that you see in pink the myosin filament, and attached to the myosin is the titin. Now, Kisa is going to be talking about the titin filament, while I will focus more on the actin and myosin here. Now, these pink myosin heads that you see here can attach to certain regions of the actin, which leads to the formation of a crossbridge. And it is these crossbridges that give rise to force, which is part of the force that you see in your whole body scale that lets you interact with the world. Okay, so that was a little bit of physiology about the crossbridge. But really, we want to be able to model this crossbridge in your computer. So I've broken this down into 4 steps. The first step is we simplify the crossbridge. Then we count the number of attached crossbridges, because really it's only the attached crossbridges that can generate force. Then we calculate the force from a single attached crossbridge, and then we add the forces from all of the attached crossbridges. Okay, so here's the complicated crossbridge. And we basically assume that this is a one-dimensional spring that can only exist in one of two states. So the first state has the actin and myosin filaments detached, and the second state has the actin and myosin attached and generating force. This means we need to count the number of crossbridges in this attached state. To do this, we assume that the actin and myosin are continuously transitioning between attached and detached states, and that transition is governed by this rate equation shown here. Now, the rate equation consists of K1, which is your attachment rate, and K2, which is your detachment rate. And this K1 and K2 themselves are a function of the crossbridge length. So what to take away from this figure is that, say, a crossbridge is at a length of minus 2.5. That means that if it is detached, it will attach at a rate that corresponds to the K1 as shown here. And if it's at the same length, but it is attached, then it will detach at a rate as given by K2 over here. Okay, so now once we know the rate at which crossbridges are attaching over time, we can count the number of attached crossbridges at a single time point using a numerical integrator. Okay, next, we want to calculate the force from a single attached crossbridge. And like I said earlier, we've assumed that our crossbridge is a spring, which means we assign it a crossbridge stiffness. And then we want to measure the length change of that spring. So if we go back to the crossbridge schematic at the top, we see x resting is the initial length of the spring, and then we know the length of the crossbridge at any given time point, which is xi, and subtracting the two gives us your delta x, which is the change in length of your spring. And given the stiffness and the change in length, we can calculate the crossbridge force. And we'll also note that your rate constant that I showed you earlier is actually a function of delta x, that is a change in length of your crossbridge. Okay, now that we have all the parts, we basically add them together to get the forces from all of the crossbridges, which is your total force. Okay, so that's how you model the crossbridge, but how do you actually run a simulation that you want to do in your experiment? So again, I'm going to simplify this into 3 steps. The first step is selecting a rate function. By that, I mean that you really choose the shape of your K1 and your K2 as a function of delta x. Now, there's a lot that goes into how you decide this. You would ideally like this to be completely based on experiment, but experimental data can be limited, in which case you might have to make some hypotheses. And as Taylor mentioned earlier, there is a second part to this workshop where I'll go into how you might go about choosing the right rate function for your purposes. Okay, once you have selected a rate function, you generate a protocol for whatever you want to simulate. And the protocol involves 3 kinds of inputs. So the first input is your time, and then you input the activation, and then the length. So here I have an example trial where the muscle is activated to a certain activation, and then during the rest of the trial, it is stretched from one constant length to another. And then you simulate each time point and generate the outputs. So that involves moving each crossbridge by the input length to calculate the delta x for each crossbridge. And then for every delta x, you count the fraction of attached crossbridges, which gives you this output, that is a distribution of your attached crossbridges. And then finally, you sum the force across all of the delta x to get your total force. And here is just the same thing, same trial in greater detail. So what I'm going to do is I'm going to walk you through what the crossbridge looks like at each time point. So here, this is how the crossbridge distribution looks at the initial constant length. And then you stretch it, and you see the crossbridges move towards being stretched. And they keep moving towards being stretched, but the distribution changes throughout the stretch. And at the new length, you see the crossbridge is coming back to a delta x of 0. So an interesting point is if you look at the first distribution and the last distribution, most of the crossbridges are at delta x of 0, which is because they're at constant lengths. But then the distribution does look different, and that is where your force due to different lengths comes from. Okay, so that was all about how you work with the crossbridge model. And here I have some results. But before I go into the results, I just want to mention that there are a lot of features to the crossbridge model. And for clarity and sake of time, I try to focus on the basic features that I would need to use on an everyday basis to generate a simulation. But there is one key feature which I left out, which is the activation dynamics, which I've added in here. Very briefly, the activation in the Crossbridge model is in the form of calcium concentration. And based on the calcium concentration, the model determines the number of actin sites and the rate at which the actin sites become available. And you need the actin sites available for the myosin heads to bind to it. Okay, so now the results I show you are based on this model. And we find that the steady state forces emerge from the Crossbridge properties. So here is the force length curve as simulated by the Crossbridge model, and this is the force velocity curve from the Crossbridge model. And below are the other data from literature. Now, if you recall, I showed these curves much earlier as well as being foundational to the Hill muscle model. Now, moving on to some transient forces. Again, this is a figure I've shown you earlier. So in the bottom row, you have a muscle fiber length change as a function of time. And at the top, you have the corresponding force changes. So in red are experimental data from a single muscle fiber. And in blue is the model prediction. And I'll again orient you to some key features. The first feature is immediately at the beginning of the first stretch, you see that the force rises up pretty steep. So if you focus on the red line, it goes up pretty high. And then the force continues to slowly increase, but not as fast. And then on the second stretch, the force does not go up as much. Now, as you can see, the model does have transient force features, but does not capture all of them. For example, while the force goes up really quickly, it also drops down, which we do not see in the data. Now, there are many ways you can improve the model, and I'm going to go in and show you a really complicated model. So the idea here is to just show you what's possible with the Crossbridge model and hopefully stimulate discussion, and we can go over this more later. But we don't need all of these states for the data that I'm talking about. So there are 5 states that we care about. The first is the detached state, and then there's the attached state, which I've been talking about throughout this presentation. And then you have the activation dynamics that I just introduced. But now I want to introduce 2 more states. One is the super relaxed myosin. So in a muscle, this is observed when there are some myosin molecules that are unable to bind to actin, but then become available as the force increases. And then there's another state, which is the forcibly detached state. And this happens when an actin and myosin molecule are bound, but then mechanically ripped off. And when this happens, the myosin molecules just immediately want to go back to being bound. Now, there's a reason for choosing these 2 states in respect to our data. It is that if you go back and look at the force traces up here, and you see the blue line drops down. And we don't want that to happen, which means we want more myosin heads to be cycling to increase the force. And these 2 states do that. So super relaxed myosin really brings in more myosin states as the force increases. And this forcibly detached myosin basically puts the detached myosin heads back into cycling. And now here are some results using that more complicated model. And I guess very quickly, what you see is now the red data, experimental data, matches much more closely to the blue model. So this is just an example. And also to show another figure is if you use the same model and the same rate functions, nothing changed. Here is what the model predicts for a completely different trial. It doesn't match very well, but it does capture the broad features. And like I mentioned early on, I'm mostly focusing on broad key features for now, but I think Kisa and Taylor will go into more rigorous metrics of how we might want to evaluate fits. Okay, so with that, I guess I just want to tie back to my earlier slide, where I would eventually like a crossbridge model that can predict muscle force, energy, and sensory signals. So I mostly talked about a crossbridge model that can predict force. But because we actually have crossbridge states and we are measuring transition between states, we can also count the number of ATPs going between the transitions, which gives us the energy consumed by muscles. And we can also predict sensory firing, because muscle spindle, which is an intrafusal fiber, is really a different type of muscle fiber, which we get by varying the rate functions that I said I'll talk to you about. So with that, yeah, so I was trying to teach you how a crossbridge model works, but the work in here is really involves a lot of people, and this is my acknowledgement to all of them.
Well, thanks, Sarabi. That was so interesting. And you introduced a lot of concepts that I'll be talking about too. So today I want to talk about predicting in vivo muscle force during perturbed locomotion. And I'll talk about both hill type and Titin-inspired models. And I really like the learning objectives, so I'm not going to have an outline anymore going forward. So the learning objectives for my talk, I want to talk a little bit about why we would want to model unsteady perturbed locomotion. And then I want to talk about hill models and Titin-based models, their parameters, assumptions, and how we might apply them to perturbed in vivo locomotion. And then finally, I'll make some comparisons across models and talk about different ways that we can compare model performance when we run the models on the same data. So it's been a really wonderful experience for me to be collaborating with Monica Daly on her beautiful perturbed locomotion experiments in vivo guinea fowl running on a treadmill. And these experiments, I think, have really taught us a lot about how muscles really function during dynamic movements. So these, and Yannick is going to talk quite a bit more about how you collect data for experiments in her talk. But briefly, as Sarabi mentioned, for muscle models, we have a time input, activation input, and a length input. And actually, to validate or test the models, we also need to know what the in vivo muscle forces are. So in these experiments, a single muscle is instrumented with sonomicrometry crystals to measure the muscle fiber lengths, and there's a tendon force buckle placed on the tendon to estimate the in vivo muscle forces, as well as EMG electrodes to give us a sense of the activation. And one of the things that you can see from some of the data down here is that what we particularly tend to see in these experiments is that in the perturbed steps, there's a real difference in muscle fascicle length from the unperturbed steps and a difference in force. And actually the force and the activation can be decoupled from each other. So you can have a higher activation than the unperturbed steps and a reduced force. So it's a real challenge for muscles to predict these kinds of data. And here's just a single trial from a single individual with, I don't know, there's about 40 or 50 steps here. And you can see actually that the effect of the treadmill perturbations that you saw in the previous video is really to increase the step-to-step variation in length, activation, and force. And so here these asterisks show perturbed steps. The steps in between are level on the treadmill. And you can see that the force is sometimes much higher in the perturbed steps than in the level steps on the treadmill. But the EMG can be about average, or it can be much higher, although the force is higher. In some of the other steps, however, the force is substantially lower than in the in-between steps. And once again, we can see that the EMG activation can be average or much higher. So what's going on with this step-to-step variation in muscle length, EMG, and force? This little picture here just shows level steps running on the treadmill, and you can see that there's small variations in length and EMG and force from step to step, but they're not anywhere near as big as in the perturbed steps. But two different things can happen in the perturbed steps. The instrumented leg can actually step up onto the obstacle from the treadmill, in which case the muscles always, and we're talking about the lateral gastrocnemius muscles here, I forgot to mention that earlier. The muscles almost always produce much higher force than average during the steps up. And sometimes they have higher EMG and sometimes they have average EMG. And you can see that there's a perturbation in the muscle fiber length compared to the level steps. When the birds step down, that's when you see the much lower muscle forces. And sometimes these lower forces can occur with average EMG or with much larger than average EMG. So basically the birds may see the obstacle coming and say, uh-oh, I better activate my muscle more. So anticipatory change in the motor control, but the force that comes out of the muscle can be quite different. And so the main point here is that these obstacle perturbations really decouple the EMG activation of the muscle from the force that it produces. And one other little piece of data I just wanted to give you before we go into the muscle modeling is that as a result of the legs stepping up and stepping down and there being level steps, we get this real decoupling between here. These traces show for like a large number of steps, like every step in the previous slide. We see the EMG as a function of time. So this would be the EMG onset is 0. And then if we look at when does the muscle force start, you can see that there can be anywhere from, as short as like maybe a 25 millisecond delay to as long as 120 millisecond delay. With the larger the force, the shorter delay, and the smaller the force, the longer the delay and this is going to really get to where we start to talk about activation dynamics, which Surabhi mentioned previously. However, now if we look at, instead of the EMG onset time, we look at when the foot hits the ground, it turns out that there's a much lower variation from trial to trial in when the foot hits the ground. So length and velocity transients associated with foot contact seem to be having a very large effect on muscle force production. And before we go on, and we'll talk about the limitations of the models, but I wanted to talk really quickly about the limitations of the in vivo experimental data as well. So, so in these in vivo experiments, we do have the force measured from a common tendon to which the lateral and medial gastrocnemius muscles attach. So we're basically ignoring the force contributed by the medial gastrocnemius. There's also noise and potentially crosstalk from other muscles in the EMG signal, and we can use a large number of different filtering methods to filter the EMG. Turns out that doesn't matter too much for the muscle models, but it's definitely something we need to think about. And then for these in vivo experiments, and Jannika's working on rectifying this situation, we don't really know what either the optimal muscle length or the maximum isometric force are for these guinea fowl muscles. So we have to make some assumptions about what those might be. So, so that's the data, and I think they're really challenging to predict for any muscle model. But now I'm going to go through and apply a hill type model, tell you a little bit about the hill models that we coded and used, and then how we applied them to in vivo perturbed locomotion. So Surabhi really nicely introduced the hill type models, and typically when you see them, they look like this. They have a contractile element. Sometimes the parallel damper that represents the force velocity relationship is included, and then they typically have a series elastic element and a parallel elastic element. But what often isn't illustrated is a very important part, which is the activation dynamics. So the activation dynamics part of hill type models takes a stimulus and converts it into a force. And typically it uses like the isometric twitch and titanic fusion properties, the rise time and the fall time of a single stimulus to convert the stimulation, for example, change in EMG voltage into a muscle force. And in hill models, activation dynamics can be first order, second order, or third order models. So the first order model includes 4 parameters. The first is like a scale factor. Surabhi talked about this in the Crossbridge models as well. So we have a scale factor that relates, for example, a given EMG amplitude to a maximum muscle force. We have a time delay that relates to excitation contraction coupling. So the delay between when we stimulate a muscle and when it produces force. And then we also have an activation time constant that describes the rate of rise in force, and a deactivation time constant that describes the rate of deactivation. And in different instantiations of the Hill model, we can have a second order activation dynamics, which takes the derivative of the first order activation and deactivation time constants, or third order models that take the derivative of the first order time constant. So we can have anywhere from 4 to 8 free parameters just in the activation dynamics. And then the muscle mechanics part of the Hill models, the force length and the active and passive force length and the isotonic force velocity relationships, are multipliers on the force that's generated from the activation dynamics to either increase the force if you're stretching the muscle, or decrease the force if the muscle is actively shortening, or modulate the changes in length based on the passive and active tension. So let's go ahead and try and apply the third order Hill model to some in vivo guinea fowl data. So hopefully these data are starting to look familiar to you. So here we have the time step-to-step varying force. This is actually at level walking, slow walking at 0.8 meters per second with no obstacles. Here you can see the length change, and these are the activation inputs. And so actually the actual EMG is shown in orange. And in this case, we used a Butterworth filter, but we can also use a wavelet type filtering process to filter the EMG data. And in this case, what we do, and actually in a tutorial, or if we actually have a time in a later workshop to run through the models, what we do is we actually optimize on the activation dynamics parameters, and the muscle mechanics parameters are basically taken as fixed and not free. So in the third order model, we have 8 free parameters that we can optimize to get the best and highest R squared. And so you can see for this data set, the R squared value is like about 0.8. So it does a really good job of predicting the overall pattern of the time varying force. And you can see that the model doesn't do particularly well at capturing the passive force during stretch before the muscle is activated. And there is a passive tension in the model, but there's no passive damping. And it turns out actually that damping forces when you're stretching a muscle passively turn out to be fairly important as well. So I think that that's one drawback of the model, is that it doesn't predict the passive tension. It predicts the rise of force well, but it doesn't predict the peak force or the decay in force particularly well either. And some of the limitations of the model are that it requires significant filtering of the velocity input. So you can't, at least in our hands, we can't just input the length trace. We have to input the velocity separately with quite a bit of filtering. There's, as I mentioned before, no viscous component to the passive tension, and therefore relatively poor prediction of the force decay, which is due to the effect of the force velocity relationship. So when the muscle is shortening fast, it seems like the model debits the force too much. One other thing that I just briefly wanted to talk about relates back to this variable time between the activation onset and the force onset. So we can use a Hill model and we can optimize the activation dynamics to fit the rise in force for a single step or a couple of steps with a very high R squared. We could, choose any particular step, in this case, a step up onto an obstacle. But now if we take those same activation parameters, dynamics parameters, and apply them to a normal step with no obstacle, you can see that the activation dynamics is quite different, and it doesn't give a good fit to the different types of steps. And one of the reasons why the activation dynamics, if you model the activation dynamics, it doesn't apply very well across steps, is because it's been known for some time that the rate of force rise and the rate of force decay depends on muscle length and velocity during activation. So here you can see this is for isometric twitch and tetanus, and you can see the force rise. And actually, what they did here is they just added a single extra stimulus between the thick black line and the thin black line. And you can see that that extra stimulus really changes the rate of rise as well. But if you shorten the muscle at different velocities or lengthen the muscle at different velocities, you can really modulate the rate of rise and the peak force that's achieved from a single twitch or titanic stimulus. So moving on to the Titin clutch model. So this is a relatively new model that we've been trying to use to, I think, achieve the same goal as Sarabi, to improve the force predictions, particularly during the non-steady state or transient perturbed conditions. That model is shown here. And it actually looks quite a lot like a Hill model. And in many ways, it is a lot like a Hill model. It has a contractile element, parallels, spring, a series spring, and we have dampers both in our model on the contractile element and on Titin, because Titin is a viscoelastic protein. But we also have this pulley that connects the contractile element to Titin. And the idea is that the contractile element, or the cross bridges, can actually rotate the pulley, which represents the thin filaments. And so this creates an in-series connection between the cross bridges and Titin. So shortening of the contractile element in muscle sarcomeres extends the Titin, and it can also, and binding of Titin to the thin filaments upon activation can change the equilibrium length of the Titin spring. So the pulley gives the Titin spring, activation dependent viscoelastic properties that depend primarily on its equilibrium length with a constant stiffness. So we have this Hill-like contractile element that basically has this very similar formula to a typical Hill-type muscle. But what we have is the contractile element force and the contractile damping force of the contractile element are equal to and opposite the Titin elastic force and the Titin damping force on the other side of the pulley. So the Titin model has 10 parameters. Actually, the ones shown in bold are the same as for the Hill model. And then in addition, it has 4 other parameters, a total of 5 free parameters. So more than a first order Hill model, but less than a third order Hill model. So muscle mass and the damping and spring constants of the contractile and elastic elements are among the free parameters. And I was really excited to see Sarabi say that her Crossbridge model replicates the force length and force velocity properties of the whole muscle. And this is also true of the Titin model. So you don't need to hard code those properties into the model. If you give the isometric or isotonic length inputs and activation in the model, you get those properties out. So what happens when we apply this model to the same data as we saw previously? There we go. So we get a little bit higher R squared, a little bit lower RMSE. You can see that there's a good prediction of the passive force, and that's because we do have a passive viscosity, not just an active viscosity in the model. And there's a pretty good prediction of the force rise and the force fall. And I'll talk just a little bit more about the fall of force in a minute. But a main limitation of this model is that these stiffness and damping constants are not well characterized across muscles or easily measured from standardized experiments on whole muscles. We're working on fixing this. And one of my PhD students, Caitlin Bemis, is currently working very hard on fixing this, but it is going to take some time. So we really don't have compared to the force length and the force velocity relationships, we don't have a good understanding of what these stiffness and damping constants are for whole muscles. But the Titin-inspired model predicts the variable A between activation and force really well. And it's hard to see this here. But the reason why it does this is all in the damping coefficient on the contractile element. So by having just simple linear damping instead of the isotonic force-velocity relationship, you can really improve the prediction of the decay in force over time using the this type of Titin-inspired model. So hopefully I'm not too far beyond my time limit. I just want to quickly talk about like 3 different ways that we can compare how well models perform when they're predicting the same data. So we can look at the prediction accuracy using metrics like R-squared, RMSE, ACA-EK information criterion, and the Bayesian information criterion. But we can also look at overfitting and biological relevance, or the contribution of different model components. So, so we already looked at R-squared and RMSE for these 2 different models on the same data. But now what we can do to look at overfitting is we can train on one data set and test on another. And if a model is capturing the real sources of variation, there should be no decrease in R-squared from the testing to the training, right? And so what we can see is, at least for these data, we actually get for this particular trial a somewhat higher R-squared. That isn't always the case for the training data. And here the testing data. So here the training had no obstacles and the test data had the obstacles. So how well can the model do if you don't optimize the parameters using the perturbed steps? But you can see that the Hill model really loses in the R-squared, and it really decreases. So something about what's happening in the perturbed steps wasn't captured in the optimization of the activation dynamics in the level walking trial. So the Titin model had a higher R-squared, lower RMSE, and actually I don't have too much time to talk about AIC and BIC today, but they actually debit the models for the number of free parameters. So even though the Hill, in this case, the 3rd order model has more free parameters, so the Titin model also does better using those criteria. In the testing versus training, the Hill model suffered more from overfitting than the Titin-inspired model. And then lastly, in terms of the biological relevance, we can ask about the modularity. So one of the things that we've done, and I don't have any hard data, but we could play with it in a tutorial. I don't have any hard data on my slide here today, is we can actually take the Hill model and optimize the activation dynamics with no muscle mechanics, or the muscle mechanics with no activation dynamics, and ask how well does the model perform. For the Titin model, what we've done is to ask what happens if you take out the pulley. So I think that there's a lot of different tests that we can do to compare models that the surface has barely been scratched previously. And it seems like there's a lot of fun and interesting work to be done ahead. And it'd be also really interesting in the future to compare these to the Crossbridge models from Sarabi as well. So that's the end of what I have to say. And a lot of different students contributed to this work, and a lot of different colleagues, including particularly Monica, have contributed data to our models. So I'll stop there and stop sharing and pass the baton to Taylor.
Thanks, Kisa. All right, I'll just share my screen. All right, hopefully you can all see my slides. Okay, so in this part of the workshop, I plan to cover, or shall I say my learning targets, I feel like I'm giving an undergrad lecture, I'll cover what muscle tendon parameters we can capture from in vivo analysis to improve our model predictions, with a focus on human models and human muscle in this part of our seminar. And then I'll showcase some different flavors of models we've used or are using to understand the mechanisms of muscle function, and particularly 1 versus 2 element models, how we can integrate these within whole body simulations, and some more recent work on integrated integrating personalized imaging data. And Kisa has really nicely, or Kisa and Sarabi have really nicely introduced hill-type models. So I endeavor to not repeat some of the theoretical side of things, but there are some key differences in the modeling approaches between our studies and the way, for example, we prescribe model parameters. And I'll try and highlight these as I go through my talk. So when we think about using models to predict muscle forces in humans, I think it's important to acknowledge that experimental tools do exist to directly measure or indirectly estimate muscle forces. And many of these tools provide really useful ways to evaluate the predictions from our models. And the only way to directly measure muscle force in vivo is invasive. So using tendon buckles, or in the past fiber optic cables, much less common in humans, but so really more suited to these animal studies that Kisa presented and Yannicka will go over. But there's also a suite of non-invasive imaging tools that have really emerged in recent years, in the past couple decades. So we've got B-mode ultrasound, which stands for brightness mode ultrasound. This enables us to visualize the strains in muscle and tendon. And with knowledge, for example, of tendon strains and tendon properties, we can begin to estimate forces. There's also shear wave elastography, another ultrasound technique where we can estimate a tissue's mechanical properties via inducing shear waves in the tissue and then measuring the velocity of these propagating shear waves. So the faster the shear wave speed, the stiffer the tissue. And more recently, shear wave tensiometry, or tendon tapping, which couples a mechanical tapper with two accelerometers to measure the speed of shear waves in superficial tendons. But limitations do exist like any other methods. For example, the muscles we have access to, typically superficial muscles, and extending some of these techniques to quantify forces during dynamic tasks really still does remain a fundamental challenge for the field. And importantly, these are estimates. So the other way to get at forces, and the theme of this workshop, is to use muscle models to predict them. And hopefully, by this point, we all agree that one of the most common models, not necessarily the best model, but one of the most common models to predict forces is the hill-type model. But like any model, it's not without limitations, particularly in predicting perturbed or non-steady muscle forces. And although widely used, hill-type models are phenomenological, meaning that the contractile component, the series elastic component, capture many aspects of force production typically attributed to our muscle fibers and elastic tissues. But the contractile element, the series elastic component, don't have direct anatomical analogs in animals and humans. And this can make comparisons and model evaluation often challenging. Also, importantly, hill-type models, or the traditional hill-type model, doesn't account for the internal mass or inertial properties. They're massless. They don't account for the fact that our muscles change shapes. They're the multidimensional nature of our skeletal muscle. They typically represent the contractile element as an average scaled up fiber, and they don't account for history or length dependent effects. So certainly some limitations. So what type of information? I think it was important to sort of talk about some of the information that we can integrate within these models as inputs that we can capture from in vivo non-invasive analysis. So focus primarily for just these few slides on imaging data that we can collect in humans, as Yannicka will focus more on the in situ ex vivo animal parameters. So first is maximum force generating capacity of a muscle, or Fmax. And in humans, we can measure the volume of a muscle. So we can do this using 3D freehand ultrasound, which essentially combines motion capture technology and B-mode ultrasound to stitch together images and create a mesh to determine muscle volume. We can also use perhaps more of the gold standard is the magnetic resonance imaging, so MRI, and segment the slices to again create a 3D mesh. And these two methods compare quite well. They act in the same way, and they provide us with an estimate of muscle volume. But that's only volume. And if we want to estimate or determine the maximum force generating capacity of our muscle, which is important for these models, as Kisa mentioned, we really want to know the physiological cross-sectional area, or PCSA. So now we need to integrate knowledge of fiber length. So again, we can use B-mode ultrasound. Here we're looking at an ultrasound video of the ankle plantar flexors just during a plantar and dorsiflexion contraction. But B-mode ultrasound still only provides two-dimensional measures. and muscle is 3D. So new tools have emerged to measure the three-dimensional length of muscle fibers, particularly using diffusion tensor imaging. But to date, this is still limited to at rest. But remember that if we want to estimate Fmax, it's not just any muscle length we want, it's the optimal fiber length that we really want to be able to measure. And a variety of different approaches have been used to estimate optimal fiber length, which are often coupled with determining the shape of the force length relationship. So for example, some have mapped out the active force length relationship for individual muscles, done so at different activation levels. You see that L naught shifts with activation level, but the shape of the force length curve, at least in mammals, appears to be fairly conserved. So if we do have knowledge of L naught, we're pretty set. And this isn't trivial, we know, and we know our models are quite sensitive to this parameter. You can also use shear wave elastography to capture information about the shape of the passive force length relationship. So what you can see here is the stretch of the ankle plantar flexors through a passive dorsiflexion, and we can see shear modulus and index of force rise over time. That the stiffer the tissue, the faster shear modulus would rise in this particular scenario. If we're interested in using hill-type models to estimate force during dynamic tasks where muscles are undergoing rapid shortening and lengthening, then we also likely care about the force-velocity relationship. And this likely becomes more important during rapid changes in muscle dynamics. I'm thinking back to Keith's step-to-step differences. And again, we can use imaging combined with dynamometry, this time using a series of typically isotonic contractions. And although we have good evidence that the maximum intrinsic speed, or Vmax or V0 changes with fiber type, the shape of this curve seems to be fairly well conserved across mammals. I think a more unanswered question is how well do force velocity properties scale from single fibers or bundles of fibers, where we typically collect this data in animal studies, directly to whole muscles. And I think this remains a little bit of an unanswered question in the field. So that was very muscle centric. And of course, our muscles connect to important elastic tendons, and we can combine measures of tendon excursion via tracking the position of the muscle tendon junction, which you can see here, with estimates of force via strain gauges or dynamometers during isometric contractions. And then we can generate these types of curves to estimate tendon force length properties. But some things we need to consider is how we should fit these data. Should it be with linear, nonlinear curves, two piecewise linear curves? And particularly, where does the toe region end and the linear region begin? And this is important because models are really sensitive to this value of tendon slack length. So essentially the length of our tendon at which muscles begin to rapidly produce force. So not trivial to collect these data. And I still think there's a lot of work to be done in this space. Okay, so that's a little bit of a very quick introduction to the experimental side of things. So now, how can we use this information within models to predict forces, and more broadly to understand sort of mechanisms of muscle function? So I'll cover three particular things that my group's worked on. So using traditional and two-element hill-type models, integrating this within whole body simulations, and more recent work on integrating personalized imaging data. So here's just an example of the types of experiments we can perform to inform and drive these models, but also to validate or evaluate the predictions of these models. And we often use cycling. It's a really nice paradigm because we can uncouple load and speed. And this is unlike walking and running. If you get someone on an instrumented treadmill to walk faster or run faster, they hit the ground harder, and ground reaction forces increase. But by changing the gears on the bike and asking individuals to pedal at different frequencies, we can uncouple these. So during these particular experiments, we used B-mode ultrasound on both the muscle belly as well as the muscle tendon junction to allow us to capture muscle fascicle lengths and tendon length changes. Another beauty of someone being seated on a bike rather than moving freely on a treadmill is just related to sensors and the amount you place on them. We can couple this with surface electromyography to understand muscle activations, motion capture to get at joint kinematics, and crank reaction forces, which are essentially the analog to ground reaction forces on a treadmill. And we can combine this with muscle models or whole body or lower limb muscle models to get at some parameters we can't measure, like, for example, time varying moment arms in these muscles. And just a quick note that these are complex data sets to collect. So as a community, I think it's important, and we are getting there to share data when possible, to test and evaluate some of these models. Okay, so now that we have the data, how can we use it within these models? And so first, the modeling inputs for these this particular approach includes time-varying muscle activation determined by measuring muscle excitation using surface EMG, as well as time-varying fascicle lengths, the resulting velocities, and pronation angles. And here's just the formulation of the traditional hill-type model, the shape of the force length and force velocity relationships that are prescribed based on literature. And just a quick note coming back to some of the things Kisa discussed about excitation activation dynamics. So in this particular study, we transformed excitations to activations using a first order transfer function based on parameters from tetanic data in cats. So at the time it was the largest mammal which we had data for. It might still be, but it really does depend on the particular data that you have. And in animal studies, you typically, if you have direct tetanic data, then I think it's been shown by Sabrina Lee as well in goats that a third order does seem to do a little bit better. And a keynote I just want to make here is that one of the differences between these hill-type models and the approach used by Kisa is that we don't optimize for any model parameters. So we don't have any free parameters in this particular model. And of course, it just depends on your research question, the approach you take. And right off the bat, I want to acknowledge some limitations and open questions using this approach still remain. So it's challenging to evaluate the forces against a gold standard because tendon buckles in humans is challenging. You can use different approaches to estimate tendon forces via tendon strains, for example, using ultrasound. Also, it's known, and we and others have shown that there's alternate force sharing between synergistic muscles when you alter the demands of the task. And here we can just see muscle activation in the medial gastrocnemius, the lateral gastrocnemius, and soleus, and the relative contributions of each changes when you increase, for example, the gear on the bike. Yet we measure strains at a common tendon. So similar limitation to what Kisa mentioned. I think an unanswered question still remains is how scalable are literature values for force velocity properties, for example, across animals of different sizes, and then which model inputs need to be subject specific. So how much experimental data do we need to be collecting? And I'll get to this on my last couple slides. And then finally, how accurate is accurate enough? Okay, so let's look at some modeling results, specifically comparing the traditional, which I'll call the one element hill-type model, and the two element model. And the primary difference between a one and two element model is that in a two element model, you have a contractile element to represent the contribution of slow muscle fibers and the contribution of fast muscle fibers. And the activation that drives each of these is derived from wavelet analysis of the EMG signal. So this allows us to estimate the contributions of slow and fast muscle fibers based on the frequency content of the EMG signal. And also, the other thing that differs is the shape of the force velocity relationship for each element, which you can see here, although normalized, so you can't see the difference in maximum intrinsic speed, for example. Okay, so how do these models perform? So up first is the traditional hill-type model. So what we found is that predicted forces were able to capture the general features of our ultrasound-based estimates of force. So forces rise in the model, peaks around the same time, and fall within the model. And particularly, we found that the peak lateral and medial gastrocnemius forces predicted by the models were generally within 10% of the peak forces estimated from ultrasound images. But the R-squared varied markedly, so from 0.4 to 0.8, with the best fits during the slower speed, higher force contractions. So the models aren't able to explain all of the forces produced. And given that model performance is poorest at these high speed movements, the ones where we likely see different contributions of, for example, fast muscle fibers, we wanted to test to see whether this 2-element model that can account for the independent contributions does any better. So when comparing then the predictions from a 1 and a 2-element model, what we found was that the predicted forces are indeed more accurate for high-speed movements when the model accounts for the different fiber types and their activation in comparison to a 1-element model, although the two models do perform quite similarly during low-speed tasks. Okay, so then to test how these models perform within whole body simulations, we wanted to essentially incorporate these 2-element models within an open simulation framework. And this is work led by the very clever Adrian Lai. And so what we did was incorporate the 2-element model into predictive simulations. And the question we set out to test with this model was, does a strategy of minimizing the metabolic cost of muscle contraction, so a cost function, does this govern the recruitment, or can we predict the recruitment that we empirically measure across different scales? The two scales being within different fiber types in one muscle, and across different muscles within a synergistic group, so the calf muscles, the triceps array. And as a reminder, we don't only see variations in fiber recruitment with task demands, we also see differences in the recruitment of the soleus and the medial gastrocnemius. And this is because the soleus is a slow fibered muscle, whereas the medial gastrocnemius is mixed, so about 50-50 slow, fast fibered. So at slow speeds, the soleus is more active, which we can see here, but at high speeds, this pattern is reversed and the medial gastrocnemius is more active. So what we did is we compared the predictions from simulations that were prescribed with the cost function to either minimize metabolic cost or to minimize muscle excitation. These are both common cost functions used within predictive simulations. And what we found was that the strategy of minimizing the metabolic cost of muscle contraction, so shown on the left, was capable of predicting the recruitment of different fiber types. I don't have this shown on this particular slide, but also the different muscles that we experimentally measured. So in other words, the patterns of modeled data best matched the empirical data for the cost function on the left compared to the cost function on the right. And what this suggests is that a strategy of minimizing a muscle's metabolic cost is able to govern the recruitment or predict the recruitment of motor units within the single muscle and the coordination among synergist muscles within a limb. So this is pretty exciting for us to see this come together. So the last thing I'll discuss is a more recent modeling question we've been trying to tackle, is how much personalized or subject-specific data is required for robust models, and particularly how sensitive are model predictions to the different parameters. And we started with Fmax, and this is work led by my PhD student, Raphael Hamard, who I co-supervise with Francois Hugues. And if we think back to Kisa mentioning that in the models they're using, this is often a parameter that's estimated. I think Yannicka will talk a little bit more about how we can measure this. So what we did is we collected muscle activations and fascicle lengths, velocities in the medial and lateral gastrocnemius, this time during walking. And these are just the different patterns across individuals, where every individual is shown by a different color. And then in these same individuals, we performed MRIs and diffusion tensor imaging to determine muscle volume and 3D muscle fiber length. And then we tested different models that relied on either Fmax determined from these MRI measurements or estimated from more traditional regression equations that just use height and mass of an individual. And what we found is that inclusion of volume and architecture from scaled generic models conceals the inter-individual variability in force sharing between the gastrocnemiae when compared to models that include these personalized measures. So in other words, we lose information when we use generic scaling. But I guess the overarching question is does this matter? And it comes back to how accurate is accurate enough? In other words, can we just use generic regression equations? And the answer really depends on your research question. So in a clinical population, or if for a surgical intervention purpose, maybe not, maybe we do need to be using personalized imaging data. But if you're interested in population level changes in muscle force, for example, with changes in task demands in a healthy population, then perhaps, yes. So really, how much experimental data is needed on a personal basis to drive these models, I think is something we're still not entirely sure of. All right, so that's really what I've got for my portion of the talk. What I plan to do in our second workshop is go through some of the aspects of these models using hands-on, a more hands-on approach. So we've developed an interactive GUI to enable us to explore how changes in these model parameters influence predictions. But I'll leave that there. But before I pass the torch to Yannicka, I need to acknowledge the amazing, brilliant group of collaborators and mentors that have contributed to this and the ideas within in my research journey thus far. So thank you very much. I will stop my share and go from there.
Well, thank you, Taylor. Okay, last bit. So now it's my turn, and I will take my time slot within this workshop to talk to you all about experimental practices, applications, and limitations. We chose to end on this note as experimental data is foundational to most muscle models out there. And for this talk, I will focus on comparative experiments, meaning that I will solely cover animal experiments. Some of the techniques I discuss have a counterpart in human experimental approaches, as Taylor highlighted, but most are totally not applicable to human subjects due to their invasive nature. To highlight where experiments and models should talk to each other, and to reiterate some of the things we've heard today, I made a quick schematic. Sorry for my drawing skills. So when we have our computational muscle model, as we heard throughout this workshop, it requires input. And these parameters like force length, force velocity, but also the muscle maximum force and activation that are required in these muscles find their origin in experimental data. The required data to provide these parameters are mostly obtained through in situ and in vitro experiments in animals, which are generally very controlled experiment with a single target muscle and sometimes partially or entirely dissected out of the animal. The other aspects where experimental data can meet models is in the validation. So in vivo data can validate muscle behavior as we collect data from the muscle in live animals while they perform a certain task. These data can help you change settings in your model or adjust your parameters. So in today's talk, I'll go over in vivo, some of in situ, and a little bit of in vitro experiments. I will mostly focus on techniques and application that I've used myself. Not on myself, but by myself. First, I'll talk about in vivo experiments. During these types of experiments, animals are equipped with sensors that are implanted in and around the muscle tendon unit. So first, sonomicrometry crystals. These piezoelectric crystals measure the live muscle or muscle fascicle length with the known speed of sound in muscle. These crystals are 1 millimeter or 2 millimeters wide that you can implant in the muscle during aseptic surgeries. Depending on the research question, but most often you aim to implant the crystals along the muscle fascicles so that your measure reflects muscle fascicle dynamics. So you see also an E-shaped tendon buckle, and in detail, it looks a little bit more like this. And now I hope you all see my cursor, but this big one on the left, that is like a raw buckle. And then we have two that are here that are, so to say, ready to go, where you see this little red dot, which is the actual force transducer, and the layer that you see over this buckle is like a rubber coating we use to protect the tendon. And just to highlight, the pictured e-buckle is these are one of the biggest I've used, simply because guinea fowl are big enough to have one of these on their Achilles tendon. For smaller animals and smaller tendons, you should use much smaller tendon buckles. So for example, a white rat uses approximately half of this size. So some animals, like frogs, do not fit a E-shaped buckle on their tendon, and then a leaf spring buckle can be used. These are force transducers on a leaf spring, which are directly sutured onto the tendon. And last but not pictured piece of equipment is electromyography. So with that, you collect relative muscle activation And for that, one implants thin EMG wires into the muscle. There are different setups for this, but I'll focus on what I've used in the past. So we use thin wires, which we coil together, and we bare the ends and create an offset at the bottom. And excuse my drawing, these wires are so thin and so fine that I couldn't make a proper picture. But what you have to get away from this is that before implantation, we coil them back up and you create a very little offset between the two bare tips. But the overall beauty of in vivo data collection is that you can see live muscle dynamics during easy or complex tasks. And most of you will have seen this video before. So we see the bird encounters obstacles on a treadmill. So what you see in purple in the upper right corner is the live work clip of this bird while running on the treadmill. The traces below show the fascicle length in pink and the force trace in blue. The black squiggly line you can see in the bottom graph is the collected EMG. So treadmill experiments are easier to do with in vivo equipment, as the animal has to have a constant stable connection with all the connected amplifiers and the computers which you collected data with. However, in vivo experiments can be done in other settings. You just have to be really mindful about the connection to the animal. And that brings me to some limitations of in vivo experiments. And I will call them thinkabouts. So besides cable length, there is quite a list of thinkabouts with in vivo experiments. I'll give some highlights only, and to name a first one, a limitation of in vivo is that the amount of data that you can obtain from an animal. So we generally collect force, strain, and activation from the same muscle, but you can only equip a limited amount of muscles at a time. Most in vivo studies target one muscle, maybe two. Too much equipment makes the surgery take much longer, the implantation of the equipment really difficult, and the transducers might pick up more noise during the data collection from each other. And on top of that, you also still want the animal to do the task. Another important thing to think about is size. Bigger animals make it much easier to equip more than one muscle. It's easier to identify muscles, and it might be easier to ask to do a certain type of task. Smaller animals make it sometimes really hard to target the target muscle during surgeries, and the weight of your equipment might have a much bigger impact. So when you think about doing in vivo data collections or you read papers, please understand that most of the equipment used, and I'm talking everything post-amplifier until the animal, is self-made, except for the sodium micrometry crystals that you can make at home, but you can also buy them. Everything we use is DIY. So when you get yourself involved in vivo experiments, you will also develop soldering and electronic skills on top of surgery skills. And despite that I now work on bigger animals, some of the plugs we have to use are still really small and really hard to solder on. And that is a theme that is inherent to these type of experiments. These type of experiments are hard, but if all goes well, it is really rewarding. Here, absolutely visually displayed, when Monica Daly and myself collected, and I quote Monica here, the cleanest EMG data ever seen. And this is a quick spoiler for next workshop, and I will show some data, and that actually, if you see clean data, that is worth celebrating. Okay, so after this quick overview of in vivo, I will now move on to in situ experiments. So in a quick schematic, this is what's going on in an in situ experiment. The muscle of interest is hooked up on one end, on the tendon end or the long tendon end, to a servo motor that registers force and length changes. At the other end, in this case the femur, is braced and shouldn't move at all during this contraction. The muscle is instrumented with sodium micrometry, and during the experimental trials, the muscle is stimulated, and here depicted, I depicted the nerve cuff, which is placed on the sciatic nerve here in back. So variations on this schematic, among others, are the way you can brace the femur or the way that the muscle is stimulated. You can directly stimulate the muscle or use a nerve cuff, like in the schematic. So muscle stimulation can be varied in intensity, pulse duration, interpulse period, or train burst width, and this depends on your experiment. For today, I'll focus on the two most common in situ experiments, which result in the force length and the force velocity curve, which are used as inputs for muscle models. So here we see a force length curve obtained from a guinea fowl last summer. If you think that these are high forces, originally with our setup, you should multiply these forces by 2. In green, we see the passive force, and in purple, we have the active force. Each one of these green dots corresponds to its own active force dot, and each couple of dots corresponds to its own tetanic contraction. The difference in dots represents a change in motor length. The change in initial length determines the passive force dots, therefore. By repeating contractions around 3 to 5 minute breaks, you will populate your force length curve, which if I add the individual contractions to clarify this statement, it looks more like this, and here based on a red LG data. This graph shows the contractions of the muscle up till the point of the maximum force, represented by the dashed lines, and each color represents its own contraction. Important to note here is that these individual contractions are done in a random order, and I will talk a little bit more about that in workshop part 2. Now that we know how the force length curve are created from a single muscle, we'll talk a little bit about these beautiful rainbows. This is the underlying data that results in a force velocity curve. For these experiments, muscles undergo isotonic contractions at a predetermined length. Normally we use for these the L0, which is determined during the force length experiments, but you can do also a percentage of L0. Every color in the graph represents a different isotonic force as a percentage of maximum force. A little spoiler for next workshop, but these graphs eventually turn into a more familiar shape of the force velocity curve we all know. Despite the fact that I've already mentioned that in situ experiments are easier as they're more controlled, there are still a series of challenges that come with these experiments. So like in vivos, in situ experiments have surgical challenges. For example, the sciatic nerve is right to a major blood vessel that supplies the leg with blood that you don't want to hit. Sonocrystals have to be placed right, and you need to be careful not to damage your target muscle if you need to cut away any of the other muscles or connective tissues to let your muscle move freely. But in situs, which is not possible in in vivo experiments, is that you can adjust your implanted transducers on the spot if something doesn't seem right. One other challenge is that you can run into, especially with a stronger muscle or with a bigger animal, is clamping. And I mean clamping on either end, so on the femur end and on the tendon end. So we've had birds who broke our femur clamps, despite I use at least 2 of these in an experiment. And clamping on the other end, so at the tendon end, can also be a problem if you have a really strong animal. So when you test rats, for example, you can leave the calcaneus attached to the tendon, so you can clamp behind the calcaneus, but in birds, they don't have a calcaneus, so we had to clamp their massive Achilles tendon, and sometimes those clamps slid off mid-experiments, which meant we had to reattach, start again, and hope for the best. Another potential issue is compliance, which can vary greatly on where you clamp your tendon. And that compliance issue is less present if you choose to do in vitro instead. But a perk of combining in vivo and in situ approaches, something we did last summer in guinea fowl, is that you can leave your equipment in place, which easily cuts off 45 minutes of your in situ surgery. Additionally, it gives you the unique individual information where your subjects operate on the force length and the force velocity curve. When they do the in vivo task that you have similar information from. Additionally, in situs create a unique opportunity to calibrate your force buckles accurately while the animal is in the in situ setup, while still attached to the tendon of interest. Okay, I will go real quickly into in vitros. So in vitro experiments to obtain force length and force velocity relationships of muscle are very similar to in situ experiments. One of the biggest differences is that there's no animal attached anymore. So depending on the experiment and the muscle of interest, you can use sonocrystals or just use the length displacement of the motor as a muscle length. A clear advantage of in vitro is that it's a very clean experiment once you get the muscle out. An important variable to choose if you can do an in vitro or an in situ is that the fact that the muscle has to stay alive. As in vitro muscles are kept alive in a solution, this would not work with bigger muscles that need blood supply to stay alive and keep working. Also, if you want to do multiple series of experiments like force lengths, multiple force velocities at different lengths and force frequencies, then you have to keep your muscle alive past what an in vitro setup can provide you. So I hope you got some idea of the basic experimental techniques that researchers use to unravel muscle dynamics and to inform muscle models. But I also want to highlight that experimental data and models can provide each other with valuable insight, as all that I talked about today is by far a one-way street. As for modelers, it is of value to know where the input data comes from and what its potential caveats are of the collected data. But I hope I've given you some insights into these caveats by explaining how this data is collected. Additionally, the other way around, we can use models and simulation outcomes and validation results to guide new experimental approaches, the study of different muscle targets, or the design of alternative experiments targeting different locomotor tasks. So the techniques I listed in this talk are hard and weren't an innate skill. Instead, I'm taught and assisted by mentors and collaborators that I want to thank here. And with that, I would like to open the floor for questions and comments. So from here on, we'll all take questions and we'll open the floor to comments and remarks as well. You can raise your hand and I can unmute you, or you can use the question and answer or the chat to type out your question. If you have any specific question for a specific speaker, please indicate so. That way it's easier to navigate who should answer. And I will stop sharing my screen here. Okay, so put your questions in the chat or in the Q&A. So I have a question from Daisy Vega. How long can a muscle survive in an in vitro setup compared to an in situ setup? And Kisa, step in if you have a better answer to that, but it depends a lot on the size of the muscle. So the bigger the muscle, the quicker it will die in a solution, and some muscles are just not fit at all to go into an in vitro bath and stay alive. So also a quick remark, and again, I can share my screen again. For our next workshop, so I want to highlight that if people have comments for that, is our next workshop is in 2 weeks. And I lost my train of thought here. But we would like to hear from all of you what you want to discuss during a more interactive workshop part. So if you want to discuss certain topics or want to see certain parts in more detail, let us know in the upcoming few days and we'll try to accommodate. I've got another question. The handheld ultrasound is so interesting. Can you get 3D motion data using this technique or just 2D?
Thanks for the question, Rachel. So if you mean 3D motion data of the muscle, we can certainly collect dynamic ultrasound data at about up to 100 with these current devices, about 150 hertz. If we combine motion capture, so a rigid body, with ultrasound, then we can basically map any pixel in our ultrasound frame to 3D lab coordinate space and understand, for example, where it is relative to where the human's moving in space. But in order to go from 2D to 3D, we need to combine it with motion capture. Hopefully that answers your question.
Shall I read out the questions? I do not know who can see and who cannot see the questions. So another question is, how do metabolic cost models change as muscle models get more complicated / realistic?
So Robbie, do you want to answer or?
Yeah, I guess I was thinking who should answer because maybe it can apply to many people. But yeah, in the Crossbridge model, essentially it's on the very low scale and you're adding up ATP molecules. And as you scale the crossbridges to muscle fibers and muscles, your energy is going to scale up. But then you have similar metabolic cost, like you have the Hill muscle model, you also have phenomenological models of muscle cost. So that's a different way. Yeah, so I think that's how it scales. maybe Taylor has a different answer?
No, I think you answered that particular question. Yeah, I think it really depends on, there's different ways you can model metabolic cost in depending on which model you're using, whether it's a hill type or a crossbridge. Kisa, did you have something to add?
Yeah, I had one quick thing to add because, you know, in the Titin models, and actually I think the data show that, you know, when you stretch a muscle, you get more force for free. And so it seems, you know, it doesn't cost extra ATP. And and in fact, you can actually save ATP. So I think that there are some, you know, known facts about muscle energetics and dynamic length changes that make it more complicated to calculate the energy cost purely from the activation input.
Okay, so Robbie, there's another question for you. Are the reaction kinetics also included in the rate constants of the crossbridges?
I guess I'm not entirely sure what they refer to, but I think the short answer is yes, because you will develop the rate functions that I showed you of how the rate constants change as a function of crossbridge length is really measured. At least some of it is measured from experimental data where you do measure these reaction kinetics.
Okay, next question. Can muscle mass be considered in the crossbridge and Titin clutch models? And do you think it would be significantly affecting predictions of real life data?
You go first, Robbie, and then I'll go.
Well, I guess I was thinking, well, Kisa, you did have that very directly, I think.
Yeah.
And yes, in crossbridge model also, like, I guess I was trying to bring down to a small crossbridge scale. But like what I showed you was how you measure force at a crossbridge level, but you do want to scale them up by adding crossbridges in series. So you will want to scale, like if you want to simulate a whole limb, you will want to scale up how many fibers you have in series, and that will be dependent on your inertial properties of the limb or the body that you're interested in.
Yeah, so quickly for the Titin models, I think this is actually a real benefit of them that I didn't have time to talk about. So the ordinary differential equations determine the rotation and translation of the pulley. And so muscle mass is one of the input parameters, and it turns out that you can have all the other parameters the same, and muscles of different masses will exhibit very different behavior, particularly in the frequency domain. So this is something that we haven't been able yet to really explore in any detail, but the potential to have particularly like the extensor, flexor behavior be quite different, because typically the extensors are much larger in mass than flexor muscles, like in most terrestrial mammals. I think there's a lot to be explored there that would be interesting in the future.
Okay, another question. Are these models significantly impacted by factors due to aging, like changes in tendon stiffness, etc.?
I'll start on that. So yes, absolutely. I think it depends. One thing we do know that, for example, specific tension changes with aging, and thus the value we use for Fmax, or even the value we use for specific tension varies. So they certainly are sensitive to these parameters. And I think if the context is to use any of these types of models for aging, that the model parameters do need to be derived based on the changes in muscle and tendon properties that are known with this population. One quick comment, Stephen, about that is one of the challenges is for tendons get really tricky, because it appears that in animal models, with age, tendons get more stiff, but in humans, tendons, at least the Achilles tendon and patellar tendon, appear to get more compliant with age, so less stiff. So I think it's currently a little bit of a challenge to have models based on literature values for aging when and applied to humans when we may see opposing effects. That's just more a comment, yeah.
I got two questions in the chat. One of them is, what methods would you recommend for collecting muscle activation data in humans during dynamic activities without a treadmill or a bicycle? So humans running around in the wild, I assume.
Yeah, I mean, surface EMG, there's lots of really good wireless sensors and transmitters. So I think your only way to do so is surface EMG. The problem, I mean, if you think about using fine wire EMG in humans, it becomes challenging if they're untethered, and also it only samples the excitations from a small population of motor units around the wire. So surface EMG is likely a better approach. Yeah, that would be my my suggestions.
Okay, so for our last, no, another question in the chat. Are there any studies comparing the performance of Crossbridge models versus Titin-Clot models on predicting the same data? If not, which model would be better for which situation?
It's like there's a collaboration brewing right now.
Yeah, I can't wait to do that, Sarabi. I hope you're up for it.
Yeah, I think that's the answer to this question, really, a collaboration, yeah.
Yes. So one of the lovely things about these workshops is developing new collaborations. And so we don't know the answer to the question, which model would be better for which situation, or what are the strengths and weaknesses. And one of the things that I've been really interested in, I think forced depression is much more important than we have thought. And so, I think the ability of all the different models to predict forced depression needs to be investigated in more detail. So forced depression is really useful in motor control for getting rid of force fast. And if muscles have to relax on their own without shortening while they do it, it takes a lot longer time. But there's also other aspects. And I think even the shape of the EMG, and sometimes why we have biphasic EMG, might be due to minimizing forced depression. So so I think that there's a lot of work to do, and the future really, I think, is to develop more different models and really test them against each other on the same data.
Another question, could you comment on how the speed of simulations optimization changes with complexity of muscle model? And what, if any, challenges does this pose?
That's a great question, and Luke, and one that Kisa and Sarabi and I sort of chatted about in terms of why hill type, why every red line in OpenSim is still a hill type model, right? It's related to the computational efficiency, particularly in predictive simulations. But Kisa, maybe, and Sarabi, maybe you want to comment on how long it takes, for example, for one muscle model to optimize for your free parameters, and yeah.
So it depends on so many things. So the main data that we've modeled in Monica's are collected and again, oftentimes the signals aren't collected at the same frequency. But we have video and sonomicrometry at 10,000 hertz, up to 10,000 hertz. And then we have, up to 60 steps over, 20 seconds. And so some of those data sets are really very large. And typically the models take, so the Titin model takes almost no time to run, but more time to optimize. And the hill, particularly the third order hill models, take a long time to run because they have to calculate the second derivative of the activation in every time step. So, and it depends on how many free parameters you have, how long it takes. But on a Macintosh, laptop running MATLAB, these optimizations can take overnight. They aren't, you don't do them in real time. So it definitely does have an impact. I think, there are consequences. So you could subsample your data to make it run faster. But for example, as I mentioned before, the sampling rates can be too high as well as too low. And so you can have to smooth the data, for the hill models as well. So I think, that's something that needs to be optimized for the particular data that you have in hand. The most typical problem, though, is that you have too little data and not a high enough frame rate.
Yeah, so I guess the Crossbridge model is maybe the most complex here. And to be clear, all the data I showed now are really forward simulations. They're not even optimizing fits right now. And because it's not, you have actual states and you're integrating each time step, it does end to be kind of slow. So when you actually optimize for fits, yeah, it can be quite slow. And that is one of the reasons why maybe Crossbridge model is not as commonly used when you study like whole body movement and things like that. But yeah, so that's like the, I guess, the pros and cons of different models, where it's faster if you simplify certain things, and you might want to simplify things based on what complexity you need to capture certain behaviors.
Okay, I think that were all the questions. Is there anybody who wants to do final remarks?
Well, I'll just say thank you so much for participating, and the questions were great, and we're really looking forward to taking a more tutorial approach in the second workshop, which is going to be at the same starting time on May 24th. So if you want to come and look at some code and try to, get a more hands-on feeling for how the models work, join us on May 24th. And if you have any suggestions or ideas for what you'd like to see in a tutorial, please feel free to contact us directly by Twitter or our email addresses that will be on the first, introductory slides. So yeah, please contact us with your ideas and suggestions.
Yeah, and I was only going to follow up and say, if there's other questions that come to mind that you didn't have the chance to ask or didn't feel confident enough to ask, please do email any of us about anything. I think, yeah, we'd be more than happy to have follow-up conversations.
And yeah, I echo their thoughts. And yeah, this was really fun discussion. So thanks, everyone.
And I would like to end with the fact that this workshop is recorded and will be found on the IOMM website. And with that, I would like to thank my fellow speakers, the people in the audience, and the NSF for generous sponsoring of these workshops. And I hope to see you all in our and other future workshops. Thank you so much.
Thanks.